where we're talking about, copper or even gold. But in actual human cases, could be people are infected with many kinds of nanoparticles at one time. Maybe silica, maybe silver, copper, or even from food material, food and water. So I'm more concerned about interaction of different nanomaterials inside the biological process. And I'm afraid that this aspect has not been considered or well examined so far.
So maybe that could be an area where standards are written to try to identify test methods to look at synergistic interactions between nanomaterials and their impact on biology.
I believe so, because some of the experiments we have done, of course, in animal model, combine some nanoparticles together, and there are certain aspects that X and Y, I cannot disclose the results at the moment, but X nanoparticle and Y nanoparticle came together, exacerbate the brain pathology, while some of the combination of nanoparticles are not so dangerous. So I believe this is the time we must consider the interaction of nanoparticles among the biological system.
You want to keep things simple. Mixtures toxicology, mixtures risk assessment is extremely challenging with soluble chemicals. So you raise a good point. I think it presents a number of significant research challenges with mixtures of nanomaterials. Rosie, were you going to elaborate about it?
Yeah, I was going to say the same thing. Not only mixtures of nanomaterials, but mixing in the disease conditions or environmental, external, and internal together. I mean, how would you organize that? How would you even think about it? What model systems would we contemplate?
It is complex, but someone has to study and someone has to find what is going on. Even we can't make any standardization on those aspects, but these are the facts.
20-year full employment for toxicologists.
I have a feeling you don't mix that in, because from a regulatory point of view, safety evaluation and risk assessment is done in healthy animals. That's the basis. If you include all kinds of diseases, it might be wishful thinking, because every different type of disease may have a different effect. I think if you're working on certain diseases, like Dr. Sharma on Lyme toxicity or on Alzheimer's, then you do your specific research. If you want to do your safety evaluation for entering a market or determine an OL of a compound, it's very difficult to include disease models. And I feel that our regular system is not, let's say, sufficient to support that. We do risk assessment on healthy animals. That's our baseline, and based on that, in specific applications, diseases come into it. When you need to develop, for example, a drug delivered to the brain, and you can also look at brain conditions, but based not on the basis. And I do believe that what you doesn't need, you're dealing with particles. You now see nanotoxicology shift not also to, let's say, doses by concentration, but doses to reach the cells. So there are also in vitro kinetic patterns on terms of formulas and calculations on sedimentation rates of the particles based on size, based on density, and then people convert the doses in the concentration in the supernatant on doses on cellular level. So to what amount of particle or dose are the cells exposed that have a toxic response. And you see that shift also a little bit in the risk assessment for chemicals and in vivo. So and it happens now also in vitro systems. So those are the adaptations, including how do you bring your particles to your target cells or target organs. Toxicokinetics or kinetics in general, both in vitro and in vivo, that's one of the first things we have to do.
Yeah, so I completely agree. I think that's a great segue into that first supplement in terms of, you mentioned some challenges associated with even doing testing, whether it's in vivo or in vitro of nanomaterials and trying to assess toxicology. So can you provide some insight in terms of some of those challenges that are seen and maybe, how we overcome those challenges?
One of the challenges is, do you use a pristine particle, or do you use how it is used by a manufacturer? Because we look for very nice, narrow size distributions. Are those the materials that are used really? Or do we look into more imperfect distribution because that's the raw material that is used in our products? So that's one challenge. So as a scientist, you want to have a pure nanomaterial with as little as much as size distribution, but we do know every particle comes with a size distribution. And depending on the quality of the material, as demonstrated for nano silver, you can have a different quality. I think it was for in vitro assays, it comes with different qualities. Those are the challenges.
If I'm not mistaken, when we study in our medical schools, that FDA has the ability of always showing drugs and its contraindication. That this drug is good for this condition, but if you have some other diseases, don't take it. You should have different things. So I guess in this way we have learned in our medical school that what drug could be good for, X disease, what could be Y disease. I'm not expert of nanotechnology regulations, but I mean when we want to make any kind of suggestions or recommendation, I believe one cannot exclude some disease processes because these nanoparticles may be more harmful to certain kinds of people. And as this is true with all kinds of pharmacological drugs. So I don't believe that nanoparticles may be a different one, but this is my personal opinion. It's up to you what you make a decision.
No, I don't disagree with you, Dr. Sharma. I think it is a significant expenditure of resources to assess that. Although I don't want to jump to the final question, I know what it is since I have inside information, but I'm wondering if I think disease models are important for medical product evaluation. They are sensitive subpopulations that could be more at risk. I wondered if there is an opportunity for some computational modeling of those disease states that would help us get to places faster than looking for empirical evidence for numerous disease states for numerous particles, and could we develop some computational methods to help us in that area?
Yeah. So I think in terms of developing animal models for disease or developing computational models, I think there's some very significant challenges in validating those against the clinical condition. I think that the whole point of looking at here with risk assessment and regulation is assessing safety of the medical device and then moving into clinical trial, where you have the actual disease state which is developed over a period of years, not acutely by modification of an animal. And you have all the environmental factors in there as well, cover the disease states, and do a very controlled clinical trial looking for safety and efficacy. And seeing through our challenges some subpopulations where there are some maybe some additional diseases in place. I think trying to develop animal models for disease is a very challenging prospect, given that often you don't end up with a model which is a direct replication of the clinical disease. And doing it with computational methods is even more challenging.
Yeah, that's a good point. Thank you, Keith. So just moving on in the interest of time, maybe Dr. Hewlett, you could give us some information or give us your insight in terms of in vitro diagnostics. I know you touched on this earlier in the first question, but what are some of the challenges that you see in terms of nanomaterials with within in vitro diagnostics? It's typically consistency, reproducibility, being able to understand the nanoparticles and their ability to translate into an effective result. So what challenges do you see in terms of that?
Yeah, we've actually been looking at this in the context of a particular nanoparticle, the fluorescent material. But certainly the challenges are, the differences of variations in the starting material, it'll be helpful to know what types of things would be useful to look at in starting materials when we accept them, what are the acceptance criteria of these materials when we get them to develop our assays. The other thing is, of course, as I showed in our research, it's variations in the functionalization, which may vary depending on what material you're functionalizing and what type of nanomaterial you're using to functionalize. That's a bit of a challenge as well. And the other challenge is actually combining the physical, chemical, and functional testing. They have to kind of go hand in hand with in vitro testing because you may have a set of specifications based on 5 different methods that you're using to evaluate conjugation or functionalization. But if it doesn't translate into a tighter, CV for the assay or a very defined LOD or a very small variation around that LOD, and also the slope of the assay. If you find there's a lot of variation there, it's not very meaningful. So those are some of the challenges we've been facing. I think it'd be helpful to, since we're talking about preparations, it'd be helpful to have some sort of a standard preparation. These may be available already, but at least for the materials that are used frequently, you said silica, silver, perhaps some fluorescent particles, they're coming more and more into use these days. And I don't know if there's been any thought given to that. That might be helpful to manufacturers, sponsors, laboratorians developing assays for in-house use. Of course, people who are doing research trying to get a better assay to do clinical research.
Are you referring to reference materials?
Yes.
So, yesterday, NIST presented a talk on their